In this video, I want to show you just another nucleophilic acyl substitution, but this one happens to be in the context of a fascinating and deeply important biological reaction. And that's how your body, or any living creature, breaks apart proteins. You could think of a protein as a long chain, and each of the links in the chain as amino acids. Now, the term amino acid is referring to functional groups that are on that molecule. All amino acids have an amine, so a nitrogen with all single bonds around it. And all amino acids have a carboxylic acid, so a carbon double bonded to oxygen, single bonded to an OH. So you could see how those molecules could be called amino acids. They're defined by the functional groups on them, carboxylic acids and amines. There are lots of different amino acids. Usually, the 20 most common ones that are in your body have the amine and the carboxylic acid connected by just one carbon in between them, all 20. And the only difference between them is what group, what other thing is attached to that carbon. If, as here, the things attached to that carbon are hydrogens, then the amino acid is called glycine. If it has a methyl group, then the amino acid is called alanine. If it has a benzene ring attached to the methyl group, benzene ring attached to the methyl group, well, you can think of that benzene as a branch, and that's called phenylalanine. So alanine was just when you had a methyl group, and you have a benzene on that methyl group, it's phenylalanine, and so on. So there are around 20 of the most common amino acids there. There are also uncommon amino acids, ones where you have more than one carbon in between. Those are not used to make most of the proteins in your body. They're often used as either neurotransmitters or for very specialized hormone functions. I'll just point out one because it will be a nice reminder of naming. So here, you can tell this molecule is also an amino acid. You have your carboxylic acid and you have your amine, so amino acid. But this is different from these amino acids below because there's more than one carbon in between. In most of the amino acids in your body, there's just one carbon in between them. Here, there's more than one, and so the name for this, you have 4 carbons, 1, 2, 3, 4. So the prefix is bute, all single bonds between the carbons. The double bond that we have is part of a functional group, which will be represented in the suffix. So bute, all single bonds between the carbons, so AN, and because we have a carboxylic acid, that's the most important of the functional groups, the suffix is oic acid. Another way to number these things is with letters. In the case of letters, the carbonyl carbon is the carbonyl carbon. The one next to it is the alpha carbon, after that the beta carbon, after that the gamma carbon. Just walking down the Greek alphabet. So on the gamma carbon, you have a gamma. On the gamma carbon, you have an amine. Okay, so the name for this, which looks a little strange at first maybe, is gamma-aminobutanoic acid. That's GABA, and you may know that as GABA. It's a neurotransmitter. It's used in your brain. Another really common one that I won't take the time to draw now, but it's very interesting, and I encourage you to look the structure up for it, is thyroxine. Thyroxine is actually an amino acid that has just one carbon in between the amine and the carboxylic acid, but the R group is very strange. It's very big. It's not incorporated in most of the other proteins in your body, and one thing you should really notice if you look up the structure for thyroxine is all of the iodine in that molecule. And it's because of that iodine that in thyroxine that they iodize sea salt, because thyroxine helps to protect against goiters. Well, thyroxine is one of the hormones that sets the metabolic rate of your body, so how quickly you burn food, how easily you gain weight or lose weight. But it has iodine in it, and if you don't have enough iodine, your body can't make thyroxine, your thyroid gland swells, that's right underneath your chin, and you get a goiter. So if you haven't heard of that, you might look that up too, goiter. Okay, there's a whole bunch of stuff we could say about amino acids. I wanted to introduce you to just conceptually what they were here. So now you've seen two big classes of biomolecules. In a previous video, we talked about lipids, so fats, which are lipids that are solid at room temperature, and oils, which are lipids that are liquid at room temperature. And those are defined by their solubility. They're biological molecules that are nonpolar. Here you're seeing proteins. Proteins are made of amino acids that are defined by functional groups, amines and carboxylic acids. So that's how you can recognize these structures. One last little vocabulary note. When amino acids form together, they ultimately form proteins. But at first, they're really small chains. As a rule of thumb, if you have a chain between 1 and 50 amino acids, it's a peptide. It's called a peptide. And then if you have over 50, then it's called a protein. Now, that's not a hard and fast rule. You'll definitely see exceptions to that, but that's a general rule. So, for example, the 39 amino acid chain that's implicated in Alzheimer's disease, amyloid beta, it's between 39 and 42 amino acids long. That is called a peptide. But something like hemoglobin, which has hundreds and hundreds and hundreds of amino acids, that's a protein. Okay, well here we have a peptide, right? This small protein really only has 2 amino acids connected to it. And so far we've discussed it in terms of just sort of landmarks and names for those landmarks. Let's look at this molecule in terms of reactivity. So we'll sort of look at it using the lens that we've been looking at in the previous videos. We have an amide. It's a carbon double bonded to oxygen, single bonded to an amine. We have an amide. It's a carboxylic acid derivative, which means this can function as a leaving group. Well, where is our nucleophile? It's a weak nucleophile. It even has a positive charge right now, but it is our nucleophile, and if you were in doubt, you could look at the product. You notice that next to that carbonyl, you don't have a nitrogen anymore, you have an OH. So the OH is our nucleophile, and the place we're going to get it is from that H3O+, the hydronium. Okay. So in the first step, we'll remember, and here I'll just draw this. In the first step, you'll remember that the amide carbon, the carbonyl carbon of the amide, is not particularly electrophilic. I mean, if you have a strong nucleophile, it'll definitely attack it. But if you have a weak nucleophile, it probably won't react. It's not especially reactive because although it's bonded to an oxygen twice and a nitrogen once, that's 3 bonds to electronegative atoms, the lone pair on the nitrogen is allylic to this pi bond. And that means that there's actually a resonance structure going on here. This pi bond, that lone pair can go toward the pi bond, and the pi bond would break and go onto the oxygen. In other words, the lone pair is adding electron density around that carbon and minimizing the slightly positive charge that's there. That makes this more stable. And that's why this is used to create structure in living things. It's no coincidence that the least reactive of all the carboxylic acid derivatives is used to create the most common structure in your body, proteins. To think about the significance of proteins, your DNA only codes for proteins. It doesn't code for sugars or fats. Only codes for proteins. And so you can see how significant proteins are. Okay, one other thing to note about that, just in terms of if you're going on to take biochemistry, one other thing to note is because of that resonance structure, this is not a single bond. Both of these bonds are something in between single and double bonds, if you average the 2 resonance structures. And that means that this whole area is rigid, and it will not bend. And that limits the types of structures, or if you prefer, it increases the opportunity for rigidity in things that you build out of proteins, or things that you build out of these amino acid amide bonds. This amide bond, by the way, between the amine nitrogen and the carbonyl, is in biology called a peptide bond, but here in organic chemistry we'll recognize it as an amide. Okay, so because of that resonance, the carbonyl carbon here is not especially reactive. And we have a very weak nucleophile. In fact, what would look like a nucleophile, what we know must be our nucleophile, doesn't even look like a nucleophile at all, because it has a positive charge. So we're going to have to make it a better nucleophile by doing a proton transfer, and we're going to have to make this a better electrophile, again by doing a proton transfer. In general, you can influence the reactivity of things by doing proton transfers, taking an H plus on or taking an H plus off a molecule. So here, the slightly negative oxygen of the carbonyl can steal one of the slightly positive hydrogens of hydronium. The electrons can snap back onto the oxygen there, and you have a hydrogen moving from one molecule to another. That is a proton transfer. Oops. Transferring an H plus from one molecule to another. So what we get after that proton transfer is this hydrogen is no longer on hydronium. The electrons snap back onto the oxygen to stabilize the positive charge there, and this lone pair is now bonded to a hydrogen. Notice that oxygen now has 3 bonds and therefore a full positive charge. So what difference does that make? Well, before, the oxygen was stealing electron density from the carbonyl carbon because of its electronegativity, and it's still doing that. But now, the positive charge is stealing extra electron density. It's pulling electron density closer and closer to itself. And so, you end up with a much larger, slightly positive charge on the carbonyl carbon. You could see how it goes from being not very electrophilic to much more electrophilic because of the influence of that slightly positive charge, or that full positive charge that forms after the proton transfer. So now that that is a good electrophile, we can take our mediocre nucleophile, water. And it's not the best nucleophile. I mean, it's an okay. It's got a slightly negative charge, but it doesn't have a full negative charge, so it's not a really intense nucleophile. But you can even take this mediocre nucleophile and attack now that much better electrophile there, the carbonyl carbon. The pi bond breaks and the electrons go to the most electronegative place nearby. That's the oxygen. It goes there to stabilize the positive charge. So what we have here is a staple of any nucleophilic substitution reaction, nucleophilic attack. Okay, after the nucleophilic attack, we get our famous tetrahedral intermediate. So, this pi bond will break. The electrons that were in it become a lone pair on that oxygen, which now no longer has a positive charge, full positive charge. And then on the other side of that carbon will be the H2O that we've added. Now that oxygen has 3 bonds and therefore a full positive charge. So, this structure, you might look at this and say, well, that's a good leaving group there. And indeed, it does leave the molecule, which just brings us back to this molecule here. So these both exist in an equilibrium, and you can see that in the arrow here. But sometimes, instead of popping off, another reaction happens. Sometimes, what acted as our nucleophile is stabilized. And that word stabilized should remind you of a proton transfer. When you change the reactivity or stability of things, you do a proton transfer. So if we had another water molecule here, and you always have galaxies of them overwhelming a solution where they're the solvent, the slightly negative oxygen in the water can steal one of the slightly positive hydrogens here. The electrons can snap back onto the oxygen, and that would give you a stabilized tetrahedral intermediate. Now the other thing you'll have here is hydronium. that's formed when the water steals the H+. So we have this stabilized tetrahedral intermediate, and in the grand scheme of things, if you look back at what we're trying to do, we're trying to replace this leaving group here with a nucleophile. We've added our nucleophile now. The OH is there. So we just have to kick the leaving group in green off. Well, right now that is not the best leaving group. And we don't have a full negative oxygen that can force it off. So instead we have to turn the leaving group into a better leaving group. And we can do that. We can make this unstable, make this stable, unreactive thing that's not likely to pop off the molecule into a more reactive thing that is likely to pop off the molecule by doing a proton transfer. So you can take the slightly negative nitrogen here, very electronegative, it pulls electrons to itself, steals one of the slightly positive hydrogens, the electrons snap back onto the hydronium, and so after that proton transfer, the nitrogen gains an H+, the nitrogen will gain an H+ from hydronium. And that nitrogen now has 4 bonds, which means it has a full positive charge. And the hydronium lost the H+, gains the electrons from the bond. Those go to stabilize the positive charge there. So now that's just water. And so now, if you compare what happened, what was achieved by that proton transfer, we turned a bad leaving group into an awesome leaving group. Before, if the nitrogen left, it would have a negative charge, unstable. Now, if it leaves, it will have a neutral charge. It will take the electrons with the bond that it has on the molecule, and it'll just use that to stabilize the positive charge that it has. So we have a good leaving group. All it needs is a slight push. And the push comes from the oxygens, the lone pairs of the oxygens next door. This carbon that by the previous life was the carbonyl carbon, that's still slightly positive. That carbon is bonded to 1, 2, 3 electronegative elements. All of them are stealing electron density away from it. And so one of these oxygens, the lone pairs feel that. They want to form a pi bond there, and now they can because they can make room by kicking the leaving group off. Okay, so if you had to classify what's happening here, it's the other perennial step that always happens in nucleophilic acyl substitutions, loss of a leaving group. Okay, well, what we end up with here is, first, 2 pieces. So let me separate those. We'll have our leaving group leaving from this molecule. Oops. And then on the other part, the leaving group will have left. Okay, so these are the 2 pieces of our molecule. The electrons that this nitrogen gains stabilize the positive charge. This lone pair becomes a pi bond. Notice that that oxygen now has 3 bonds. So let me draw these in slightly more conventional ways, with the bond angles a little bit more representative of what they actually look like. On the left here we have 1 amino acid. The oxygen, the carbonyl oxygen, is just bonded to a hydrogen, which is slightly unstable. We're going to have to stabilize that. And on the right, we have another amino acid. So we have those 2 amino acids. All we need is for this water to swoop in and help us out, help us stabilize this with a proton transfer. Whenever you want to affect the stability or reactivity of things, do a proton transfer. So the slightly negative oxygen can steal the slightly positive hydrogen, the electrons snap back onto the carbonyl there, and you would have really these same two products just without the hydrogen there. So the electrons turn into a lone pair, and now what was water you have as H3O+. So notice we started with H3O plus hydronium, we end up with hydronium. It's not used up, but it does make the reaction happen. That's the definition of a catalyst. So this reaction is acid catalyzed. Now it's because this is acid catalyzed that you have acid in your stomach. Your stomach is where proteins start to break apart. The acid forces this very reaction to happen during every meal that you eat. And you end up with two amino acids separated by each other. Ultimately, what we did was we replaced the leaving group, the amine of the amide, with an OH from our acid and water. And if you zoom out, you'll see the same general structure of the reaction as we've seen before. You have the essential steps that always occur: nucleophilic attack that brings in the nucleophile, and the loss of a leaving group that makes the leaving group leave. And everything else is a proton transfer. A proton transfer in the beginning, 2 in the middle, and 1 at the end. So we've seen this many, many times now, but hopefully seeing it in this biological context gives you some sense of the significance of all of the organic chemistry that you're learning this semester. It will ultimately pay off. It will connect, it will build, it is a bridge that is connecting to the biology that you have already learned, so that your understanding of the world will be fluid and more complete. So that is the acid-catalyzed hydrolysis of an amide, just by using a dipeptide, 2 glycines stuck together.